Today, I hope to convince you that in the last 20 years, we've undergone 3 major revolutions in biomedical research, which allow people like me and my students to discover potential drug targets to treat cancer dressed in our PJs. So, what are the 3 major natural sciences that we all take in high school? Biology, chemistry, and physics. Cool. Among those, which were mathematically intensive? And chemistry. You're giving my talk. So, 21 years ago, that changed. In 1995, the genomes of 2 bacteria, Haemophilus influenza and Mycoplasma genitalia, were sequenced, meaning the letters of their DNA, A, C, G, T, were read. Now, analyzing these genomes, now the influenza genome is nearly 2 million letters or bases long. So analyzing these genomes, which are finding genes which are like words embedded in random sequence, required people with computer programming and statistics backgrounds. They required strong mathematical, computational backgrounds. As a result, computer scientists, statisticians, mathematicians, physicists, chemists entered the field of biology, given this new demand and opportunity to apply computational approaches to improving our understanding of biology and human health. In 1995, biology became a mathematically intensive natural science. This is the first revolution. Now, I mentioned that influenza genome has nearly 2 million DNA letters. Your genome contains over 3 billion. The Human Genome Project, consisting of a consortium of 20 top research institutions, started sequencing the human genome in 1990 and ended in 2003. What was the cost? Nearly $3 billion. And it took a consortium 13 years. Today, the cost is $1,000, and we can do it in a day. This dramatic cost reduction has been driven by the development of a new technology in 2005, high-throughput sequencing. These are instruments that can sequence the letters of DNA really fast and really cheap. This is the second revolution. Okay, now many of you may have heard of Moore's Law, right? This effectively quantifies the rate at which computers are getting faster and cheaper. That's the white line in this figure, shown as cost reduction over time. The green line shows the cost reduction of sequencing the human genome over the same time period. It's plummeting, much faster than computers. It's arguably been the fastest moving technology on the planet in recent years. So the first revolution brought people with computational skills in 1995 into biology. The second revolution in 2005 was development of instruments that could sequence the letters of DNA really fast for really cheap. What do you think has happened since? The amount of publicly available data has skyrocketed. Here is the growth of one major genomic data archive that currently contains nearly 3,000 trillion DNA letters, and it's still rapidly growing. These instruments have driven the success of big science genomics projects like the Cancer Genome Atlas, where teams of scientists and clinicians are sequencing the genomes of patient tumors across a wide variety of tumor types. It's also driving the success of a number of other big science genomic projects, each of which deposit massive data sets. Genomics data is also being generated by thousands of labs around the world as part of their research. So what does this mean? It means that people like me can discover drug targets to treat cancer working from home, reanalyzing publicly available data, dressed in my PJs. This is the third revolution. Now, as an example, I'm in a collaboration with a colleague and friend, Dr. Marty Mayo. Broadly, the goal of our research is to better understand how lung cancer spreads to other parts of the body, say bone, a process known as metastasis. Now, for many tumors, metastasis is what kills a patient. So we search for proteins that, when blocked, block the spread of cancer. This could significantly extend patient lifespans. So, simulating a tumor environment, Marty feeds the cells a protein. When the cells eat this protein, they change. They start out held together by junctions. The junctions retract. They're degraded. The cells change shape, and they become mobile. They can actually become more like stem cells and revert to other cell types, not just lung. So, when they travel to bone, they can become bone tumor cells, thereby seeding a new tumor in bone. This is metastasis. There are about 20,000 genes in the human genome, which ultimately produce proteins. We mapped 18 factors that reduced the list to 600 that were critical for metastasis. We then compared this list of 600 to a database of proteins that are secreted in these tumor cells or spit out, and we found one protein that the cells make themselves and eat, thereby becoming self-reliant at maintaining the metastatic state. Marty's lab tested this prediction, the importance of these proteins that the cells make in metastasis experimentally. When you block this protein, the cells still make it, but they can't eat it. This halts the spread of the tumor to other tissues. This could lead to a therapy to treat metastasis in lung cancer. So the first revolution brought people like me and my students into biology. The second revolution allowed us to map the gene control factors that took the list from 20,000 genes to 600 that could be important for metastasis. The third revolution allowed us to compare the 600 genes to publicly available data to arrive at one protein that, when blocked, blocks metastasis. I believe this will be the century of the cell. Our increasing power at making and testing precise predictions of cellular function will deliver precision medicine. Medicines tailored to your genome. Therapeutics specifically targeting an individual patient's mutations. Now, so if you're good at math and computer programming, and someone asks you, what do you want to do next? A great answer would be, become a biologist. You'll be able to discover precise therapeutic targets that revolutionize patient care working from home in your PJs. Thank you.